Assalamu alaikum. So here we will be talking about a number of techniques for the purpose of analyzing gene expression. That is how active genes are, and to estimate, determine, assess, measure the RNA levels in different samples. So let's sort of talk about these techniques. Now I can categorize these techniques into three levels of complexity. We have basic methods, and there are two of them, northern blotting and in situ hybridization. There are advanced methods, and these include real-time PCR and DNA microarray. And there is a more advanced method known as RNA-seq or RNA sequencing. So let's start with the northern blotting. So basically, this is done exactly the same way as southern blotting, except that instead of isolating DNA and analyzing DNA, here we're talking about uh, isolation of RNA molecules and analyzing these molecules. Once isolated from samples, the RNA molecules are then fractionated. They are separated according to size using gel electrophoresis. So just like separating DNA fragments, we can have different types or different sizes of RNA molecules depending on the size of the gene, right? So we're talking about large DNA molecules, and we're talking about small DNA molecules, and RNA molecules, RNA molecules in between. Then the RNA molecules are transferred onto a membrane. We talked about that before. And then we add a probe. Usually the probe is a DNA probe. Now it doesn't matter. It doesn't matter if it's RNA or DNA, but DNA is more stable, and that's why we use DNA probes. Now, you can have a combination of double-stranded molecules, by the way. So you can have double-stranded DNA, you can have double-stranded RNA, and you can also have one-strand DNA, one-strand RNA as well, hybridizing or hydrogen bonded to each other. So you can think about the different information that we can get from this technique, from northern blotting. We can get the size of the RNA molecule that we are interested in. We can get the expression, if it is expressed or not, and how much of it is expressed. So again, let's go over the procedure here. So again, the RNA molecules are separated based on size. Okay, here we go. So they're separated based on size. Then they are placed onto a membrane, and the RNA molecules are transferred to a membrane. Now, the molecules are transferred exactly at the same relative position as they appear in the gel. So we have the large molecules here and the small molecules right there, and things in intermediate are in between, right? So, and we have different samples as well. So we have sample 1, sample 2, sample 3, sample 4, and so on. So the RNA molecules are transferred onto a membrane, and then a probe is added. Like I said, it's a DNA probe usually. And whenever there is an RNA molecule, a messenger, by the way, we're talking about messenger RNA molecules here in general, okay? So, and we're talking about mature RNA molecules as well. That is, they are spliced, they may be polyadenylated, capped, and so on. Anyhow, so regardless. So if there is an RNA molecule that is complementary to the probe, that has a portion, that has a sequence that is complementary to the probe, now the probe is going to hybridize to it. And the probe is labeled. Usually radioactivity, some do fluorescence, but usually it's radioactivity. And wherever the probe binds, we're going to get a signal. Okay, so for example, right here, we get a signal as shown in these bands. So the bands of RNA look exactly like bands of DNA as well. Okay, so what we get from here is that the size, so we have samples 1, 2, and 4 are expressing this gene. They are transcribing this gene. Sample 3 does not transcribe this gene. Why? Because there's no signal. Number 2, we know that the RNA molecule is small. It's a short RNA molecules because if this is the top of the gel, the top of the membrane again, this means that the RNA molecule, the RNA fragment, is quite small. All right, so these are the two pieces of information. Well, let's look here. Let's look here at this gel right here. So let me try to zoom in. Can I zoom in? No, I cannot zoom in. I cannot zoom in. Well, the idea here is that look at these samples. So we're looking at expression of a certain gene. We're looking at the RNA level in different tissues. Okay, so what tissues do we have in here? Let's see. So number 1 is retina. Number 2 is total brain. Let's look at number 4, cerebellum. Okay, so what do we see in here? So we see that the gene is expressed in these different tissues. The band right here is more intense than the band nearby. Okay, which means that cerebellum expresses more of this gene than the temporal lobe. These tissues do not express the gene whatsoever, okay, because there is no signal. All right, so that's basically it. So these are the different information that we can get. We can get if information on whether the gene is expressed or not, what the size of the messenger RNA is, and number 3, to what extent is the gene expressed? How active is the gene? So the more intense the band is, the more expression it indicates. All right, so let's look here into different samples. So we have 1, 2, 3 samples. These are the wells. And here we have sample 1 expresses a relatively large messenger RNA. It produces a large messenger RNA. Number 3 expresses the gene, okay, but the messenger RNA is smaller. Number 2 expresses the gene and it produces 2 different messenger RNA molecules. So think about why is it that they express different types of RNA molecules? Well, if we go back to the lecture on phenomena related to transcription and gene expression, you can learn about how different messenger RNA molecules can be produced from the same gene. So we're talking about different promoters, alternative splicing, the termination of transcription, okay, and so on. So these are different reasons for why we can get different messenger RNA products from the same gene. So again, the probe binds to messenger RNA regardless of its size, as long as it's complementary to it. And remember again that before we add the probe, we separate the RNA molecules based on size. So the probe does not influence, it does not affect how the RNA molecules are, how the RNA molecules migrate through the gel. Now, how about expression? That is comparative expression. So we're comparing the levels of expression among different samples or between different samples. These samples can be placed under different conditions. For example, So for example, muscle cells plus minus potassium ions, for example, or certain endocrine cells in the presence or absence of a certain hormone, and how this hormone might change the levels of expression of certain genes. So we're doing comparative expression among different samples. So here we go. So we have three samples right here. 1, 2, 3, and this is the gene of interest. So we're looking at the expression of that gene. So the the size of the messenger RNA is about 9, let's say 9 kilo bases. Remember, it's single-stranded, right? So it's not base pair, it's kilo bases. So here we have there is an increased intensity of the band, right? So you see that number 3 has more intense band relative to number 4. Number 2 has more intense band compared to number 1. So one might say, well, sample 3, or the sample at condition number 3, there is more expression of the gene of interest because there's more intense band. Well, one might say, well, you know what? There is some sort of technical error. So for sample 1, let's say we used 10 microliter of our sample. For sample 1, we used 10 microliter. For sample 2, we used 50 microliters. For sample 3, we used 100 microliters. So one would say, of course, it makes sense that number 3 would have a more intense band because you use more material, more sample, larger amount of sample. So there is more intense band. Well, one would say, well, no, I did not do any mistake, and the proof for that is that we looked at a housekeeping gene, the expression of a housekeeping gene. A housekeeping gene is a gene that is expressed and its expression does not change under different conditions, like histones, for example. The expression of histones usually does not change because it's always expressed. The expression of actin, the expression of certain metabolic enzymes like glyceraldehyde 3-phosphate dehydrogenase. Uh, all cells need metabolic enzymes, and these metabolic enzymes, the expression of these metabolic enzymes does not change. So one would say, well, let's look at the expression of a housekeeping gene. So by looking at the expression of a housekeeping gene, we see that the expression is equal, meaning that the increase that we see in here is true. It's a real change. It's a real increase in the expression of that particular gene because the housekeeping gene, the expression of the housekeeping gene does not change, indicating that we've used the same amount of sample. We use, let's say, 10 microliters here, 10 microliters here, 10 microliters here. So there is no variation in the volume or the concentration of these samples. We use the same concentration, and this is evidenced by the equal amount of housekeeping gene messenger RNA in the 3 samples, and the increase that we see here, the increase in the intensity, is an expression of, or a reflection of, increased expression of that particular gene. Okay, so here's another example. So that's the gene of interest, and there is more intense band indicating or suggesting that there is more expression. But if we look at the housekeeping gene, we notice that the bands also get intense as well. And that means that we do not have equal amount of sample added to each well. Okay, so the increase that we see in here is not real. So that basically what it means. That's what housekeeping gene is. So we talked about different types of bloods when we analyze DNA, RNA, and proteins. We talked about the protein analysis in the biochemistry course, where we separate proteins according to size. And then what we do is that we add antibodies. So this is called immunoblotting or Western blotting. So we do SDS, sodium dodecyl sulfate, PAGE, polyacrylamide gel electrophoresis, followed by transfer of proteins onto a membrane, and we add an antibody that is specific to that protein. We talked about northern blot right now, and we say that this is a way of analyzing gene expression by looking at the presence of RNA bands, uh, RNA fragments that are complementary to a specific probe. We look at the presence, yes, no, we look at the size, and we look at the intensity as well, which indicates the level of expression. We also previously talked about southern blotting, where we analyze the sizes of DNA fragments generated by restriction endonucleases, for example. Now, the other technique is known as in situ hybridization. You know what hybridization means. Well, how about in situ? Well, in situ means in place, okay, in place. So what we're doing here is we're looking at the expression of RNA molecules in cells in tissues. Okay, so we're looking at the localization where a gene is expressed in a tissue section. And you already know that tissue sections contain a lot of cells. So in any tissue section, you may have something like epithelial cells, endothelial cells, fibroblasts, inflammatory cells, and so on. There are differentiated cells, less differentiated cells, stem cells, and so on. Now, what we do here is that we take a tissue section and we add a DNA or an RNA probe on that tissue section, and we see where the signal is coming from. So for example, right here we have a tissue section. And in this tissue section, there are different types of cells. There are the stromal cells, fibroblasts, inflammatory cells, endothelial cells, muscle cells, etc. And we have epithelial cells right here, or keratinocytes, for example. And there are immature cells and well-differentiated cells that are up here, and so on. Well, what we do is that we take this tissue section and we add a probe to that section, and the probe emits a signal. So we see where the signal is coming from which cell the signal is coming from. So we zoom in onto a certain cell, we add a probe to the whole tissue section. So we zoom in here, we see that the probe binds to the messenger RNA molecules in that particular cell, for example. Okay, so and we visualize it. We visualize where the signal is coming from. So we know exactly that that this cell expresses that gene in particular. So this is actually taken from a Drosophila embryo. This is the embryo of fruit flies, and different in situ hybridization techniques were done using different probes for different genes, and you can see the specific and pattern of expression of certain proteins related to, let's say, development, the embryogenesis process of the fruit flies. So you can see the pattern. Well, one would say, well, you know, what's the importance of looking into that? Why don't we just do immunohistochemistry? That is, why don't we look at the protein expression? Well, it turns out that sometimes you can have a gene expressed in certain cells, but the protein appears somewhere else. So for example, with in situ hybridization, we looked at the expression of this gene right here for this particular protein. So we looked at the RNA level, okay? So in situ hybridization, we're looking at the RNA level, and we noticed that the RNA is expressed in those cells that line up along the basement membrane. But when we looked at the protein using immunohistochemistry, we noticed that the protein exists in the basement membrane. Okay, so this means that these cells express the protein, and right away the protein is exported into the basement membrane. So we know that the source of this protein is not these cells or those cells or whatever, rather the source of the protein is those cells right here. Okay, so this is the importance of in situ hybridization. We're looking at the expression of genes in tissue sections, okay? And we look at the intensity as well. We look at the intensity as well. Well, remember how we talked about real-time PCR, and we said that we can quantify the amount of DNA in a sample. Well, we can do the same exact thing using on RNA, except that in order to do quantitative PCR on RNA, we first need to convert RNA to complementary DNA or cDNA. And that's exactly what we do. Okay, so we convert RNA to complementary DNA, cDNA, using reverse transcriptase. And then we do PCR. And the idea here is that the more RNA there is in a sample, the more cDNA of that RNA exists in that sample. The earlier the detection would happen using quantitative PCR. Now, this is known as quantitative reverse transcriptase real-time PCR or RTQ-PCR, okay, for reverse transcriptase, because we're converting, again, the RNA to DNA. So we're looking at the quantity, the amount of RNA in a sample. So what we do, again, we convert RNA to cDNA, and then we quantify the DNA using primers, just like PCR. And we use cybergreen, by the way. Or we can use TACMAN technique as we described earlier. So here we have different samples right here. Okay, and notice that we have 1, 2, 3, 4, 5 different samples, and this one here is detected. We are looking at the what? At the threshold cycle, the CT cycle, the cycle where the signal is detected. So right here, the RNA or the cDNA is detected at cycle, let's say 13 or 14. This one here is detected at cycle 16. Okay, what does that mean? It means that this sample right here has more RNA than the second sample, this sample right here, okay, because the signal is detected at an earlier cycle. This one right here in red, this sample has the least amount of RNA. Okay, so there's more expression of a gene, let's say, in this sample versus all of the other samples. This one here has the least amount of RNA. And so this is based on cybergreen. Okay, this is based on cybergreen, which is a chemical that binds to double-stranded DNA, and it fluoresces when it binds to double-stranded DNA. Of course, the more DNA it binds to, the higher the fluorescence, the stronger the fluorescence. And by the way, if we do comparative expression, this is the gene of interest. This is a housekeeping gene. One would say, well, do I need a housekeeping gene? And the answer is yes. You also need to do a housekeeping gene. So a housekeeping gene right here for two samples, dark blue and light blue, indicates that the cycle, the CT, the threshold cycle, is exactly the same. So we're using equal quantities of total RNA in these of these two samples. But then when we looked at the gene of interest, we notice that the signal is detected in this sample at an earlier cycle compared to this sample. Okay, and that means that this sample here has more expression, higher expression of that gene compared to that sample. Okay, because the RNA or the signal is detected at an earlier cycle, and there is no technical error in terms of the starting material, because when we looked at the housekeeping gene in these two samples, the signal is detected at the same threshold cycle, so we're using the same amount of RNAs as the starting material. So I hope that you got the idea. And quantitative reverse transcriptase real-time PCR was done to detect the coronavirus during the COVID-19 pandemic. And what happened was that they would take the sample. This is how it went. They would take a swab from a person suspected to have COVID-19, and the RNA, the viral RNA, would be extracted from that sample, and then it would be converted to cDNA and then amplified. That RNA would be amplified. And in order to convert, or in order to confirm that this was a coronavirus, three genes were amplified all at the same time. So multiplexing was done. Remember, multiplexing with PCR, multiplex PCR, which means that several genes, several DNA molecules were amplified at the same time from the same sample or in the same sample. So right here we have amplification of three coronavirus genes at the same time, and they're all positive. And this indicates that the person has a coronavirus. So all of the techniques that we talked about before basically analyzed uh, one or few genes, northern blotting, in situ hybridization, even real-time PCR. Well, one would say, well, I want to look at all expressed genes at the same time. I want to have a clear, big picture of what happens in a cell. Remember that we have, let's say, 20,000 protein coding genes, and we do not expect all 20,000 protein coding genes to be expressed in all cells. Rather, there are cell specific or tissue specific genes, like insulin, for example, or neurotransmitters of some sort, and so on. And that's why the sciences became big, and there is a need for bioinformatics and data analysis. So by looking at the whole genome, at the whole DNA in a cell or a tissue or an individual, we're doing what is known as genomics, the science of genomics. One would say, I want to look at the expressed genes at the RNA level. I want to look at the transcriptome. And the transcriptome is the total collection of transcripts, the total collection of RNA in a cell. And that's the science of transcriptomics. One would say, no, 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 I want to look at the proteins, all proteins expressed in a cell, all proteins that exist in a cell, and that is the science of proteomics, which looks at the whole proteome. Proteome is the total collection of proteins. One would say, no, no, I want to look at metabolites. I want to look at glucose 6-phosphate and pyruvate and lactate and fatty acids and all of that. And that is the science of metabolomics. So here I want to focus on transcriptomics because we're talking about RNA analysis. Okay? And here we want to talk about microarrays. Array means مصفوفة. That is a lineup of RNA probes attached to a solid surface. Usually it's a glass microscope slide or a specific chip of a specific platform or surface. And this surface contains many different probes, and each spot contains multiple probes, identical probes that are specific for a specific gene. So you can look at, YouTube videos or the animation videos that I uploaded on Teams, or you can, look at these links if you want. So here's the idea behind it. Okay, so here we have a probe that is attached to a solid surface on a certain spot. Okay, and then when we add a a DNA or RNA to that surface, if the DNA or RNA is complementary to that probe, it's going to bind to it. So it's based on hybridization. And the signal comes out of this solid surface, of this location, of this position right here. Okay, so that's the idea now. So here we have the solid surface. Let's zoom in here. Let's look at this. You notice the dots? Well, each dot, so let's zoom in here further. There are 4 spots right here, 4 spots. And here we're talking about millions of spots, by the way. And here we're talking about, again, millions of spots right here. So each spot contains multiple probes of the same type, and these probes are identical, are complementary to a specific RNA molecule or DNA molecule. Okay. Now this one here is multiple probes for another specific gene. So that's gene A, gene B, gene C, gene D, etc. So we can have all 20,000 protein coding genes right here. In fact, since we have non-coding RNA genes, we can have hundreds of thousands. We can have like something like 120,000, well, that's hundreds of thousands, 120,000 different probes. So, and each one of them, I know what each spot is specific for, what each spot detects. Okay, and this is, by the way, the chip. That's how it looks like. It's really as small as this right here. Okay, so this whole thing exists right here in this box in here. So here's how it's done. We take 2 different cell types: normal, cancer, normal, diseased, treated, untreated with a certain hormone, let's say. We isolate the messenger RNA. We convert it to cDNA and we label the cDNA molecules. And then we add this whole thing to the DNA microarray. And each one of these cDNA molecules is going to bind to a specific probe that exists on the surface at a specific spot. So I know that this spot is specific for gene A, this is gene B, this is gene C, gene D, etc. Okay, and a signal comes out of every spot. And the signal, the intensity of the signal, indicates the level of expression, the amount of the initial RNA molecule, because the RNA is converted to cDNA. So if you have 10 messenger RNA molecules, you're going to have 10 cDNA molecules. If you start with 100 messenger RNA molecules, you're going to get 100 cDNA molecules. And as a result, you have 100 cDNA molecules binding to a probe at a specific spot, or 10 cDNA molecules bound to to probes on another spot. Of course, the 100 cDNA molecules are going to emit, they're going to generate a stronger signal, and that means that there is more expression of one gene versus the other. So you can look at the colors right here. So the darker the red color, the higher the expression. So the darker the red color, the more cDNA there is in that for that particular gene, the more messenger RNA, okay, the higher the gene is expressed. Now, if there is a white signal for, let's say, or no signal, that is, it means that there is very little expression or no expression whatsoever. So we can compare the intensities of the signals coming out of from every spot in these two different samples. Okay, so of course there are normalization techniques as well. That is to looking at housekeeping genes again to make sure that when we do comparisons between the two samples, we can indicate if there is a true increase in expression or if it's just a technical error. So you can read the details in this slide right here. So that's the idea. So here we have just zoom in into the microarray. So you can see the different colors that can be generated. Now each dot right here indicates a specific gene, a probes for a specific genes. Okay, so you can see how meticulous this technique is. Now, and the last technique I want to talk about is RNA-seq, which is quite advanced. It requires a lot of bioinformatics analysis. So with RNA-seq, what we do is that we take the messenger RNA molecules from a certain sample, okay? And of course, we have different amounts, different numbers of molecules of these messenger RNA molecules, and they are converted to cDNA, complementary DNA. Now, so if you have 4 messenger RNA molecules, we're going to get 4 cDNA molecules. If we have 1, we're going to have 1. If we have 2, we're going to have 2 cDNA molecules, and so on. And then these cDNA molecules are sequenced, okay, doing the next generation sequencing, the high throughput sequencing method that I talked about, where we can sequence so many DNA molecules all at the same time. So you can go back to the DNA sequencing lecture. And the number of sequencing events that take place for a particular cDNA indicates the amount of cDNA that exists in that sample. So this cDNA molecule, for example, would be sequenced 4 times. This one here would be sequenced just once, and so on. So not only that we know the amount of a cDNA molecule, that is the amount of messenger RNA, or how good a gene is expressed, we also know the sequence of that RNA molecules. And that's how we found out that 75% of the human genome can be transcribed by just doing this sequencing for everything that exists in a sample. So with RNA-seq or RNA sequencing, we know the relative amount of cDNA molecules or messenger RNA molecules in a sample, and we know what the sequences are. So even though we may not know the exact nature of that gene, the exact identity of that gene, we can know its sequence, and we can go back and search the database, the human genome database, to see where it exists exactly, on which chromosome and whatever, and we can study it further. I hope it's clear. So there are differences between RNA-seq and microarrays. RNA-seq, with the RNA-seq technique, we can characterize novel transcripts, the genes that we didn't know about. We can identify splicing variants, by the way, and that's important. We may not be able to do that with the microarray, but we can do it with the RNA-seq, and we can provide the expression pattern, the expression levels of all transcripts in a sample. So microarrays have their own limitations, and RNA sequencing can solve these limitations, can overcome these limitations. I hope it's clear, and I hope you've enjoyed it, and that you understand these different techniques. Allah ma'akum. Right. So let's talk about something different here. Let's talk about how we can analyze transcriptional regulatory sequences. Now, we talked about DNA regulatory pieces of DNA, like promoter regions, for example, or enhancers. These are binding sites for specific promoters, for specific, specific regulatory proteins. General transcription factors, specific transcription factors, like cyclic um, response element binding protein, for example. It binds to a specific promoter proximal element. There are specific proteins that bind to enhancers. Okay, these proteins cannot bind to other enhancers. Well, so let's say that I want to study how good a gene is expressed in a certain cell type at certain conditions. I don't know, in the presence of hormone versus absence of this hormone, let's say. In the presence of light versus in the dark, and so on. So we want to study how good, how efficient a promoter is at certain conditions in expressing a certain genes. Now, note, I am not interested in studying the gene itself. I'm interested in studying the promoter of that gene, not the gene itself. Okay, but I want to study how good a promoter is at certain conditions. So we can use DNA cloning, the creation of recombinant DNA, to do so. So what are the different transcriptional regulatory sequences? Well, here we're talking about the promoter, we're talking about the promoter proximal elements, we're talking about enhancers and silencers. So we have a gene, we have transcription starting point right here, right? And right before the transcription start site, we have the different regulatory sequences. We have the promoter, promoter proximal elements, enhancers, and so on. Now, we want to study this, let's say we want to study this region right here. Now, note that we can have silencers instead of enhancers, okay? So, let's talk about something different now and see how we can combine all this knowledge together. So we take advantage of an enzyme that is expressed in fruit flies. Okay, so fruit flies are these insects, and their butts glow, right? They light up, creating this beautiful scene at night. And the enzyme is called luciferase. Luciferase. Okay, and what it does is that there is a substrate called luciferin, and this luciferin is cleaved or converted by the luciferase enzyme into oxyluciferin, and this is the molecule that lights up. So I don't care about the reaction itself. All I care about is that we have this enzyme and it cleaves luciferin, creating a product that lights up. Okay, so how can we take advantage of all these tools, molecular tools? So that's what we do. Okay, we create recombinant DNA using these plasmids. So we have a plasmid, bacterial plasmid, and we take the fruit fly luciferase gene and we add a promoter that we want to study. So we're not using the promoter, the original promoter of the luciferase gene, right? So we have it under control of a promoter of interest. So let's say that we have the luciferase gene be under control of the enzyme, the hydroxylase enzyme, the dopamine hydroxylase enzyme, for example, the promoter of the dopamine hydroxylase gene. Okay, and so we create this recombinant DNA. and we transfect, we insert the plasmid into a human cell, a nerve cell, let's say. Okay, so now this nerve cell has its own DNA of interest. Okay, now we can place these cells under different conditions. Okay, and we can then study the expression of the luciferase gene, which is under the control of the promoter of interest, under these conditions. In the presence of light versus dark, in the presence of a hormone, in the presence of a certain neurotransmitter, stress, you name it, okay, you name it, it doesn't matter. Now, we can then add luciferin. And luciferin, if luciferase is expressed, it's going to convert luciferin to this molecule that lights up, the product that lights up. Now, the stronger the signal, the stronger the light, the more the gene is expressed, or let me say, the more the gene is, the higher the activity of the promoter is, the more the gene is expressed. The more luciferase is in the cell, the more light is gonna be produced. Okay, so that's the idea. So we place these cells in different conditions. In the presence of hormone versus absence, neurotransmitter, yes, no, stress, yes, no, and so on. And we measure the amount of light. And that gives us a signal, an idea of how this promoter is regulated at different conditions. Okay, that's one thing. Something else is that we want to study this regulatory sequence. Totally different idea now, okay? We want to study what we know that this region contains regulatory sequences. Promoter, proximal element, promoter, enhancer, whatever. I want to study the position, the location of all of these regulatory sequences. The exact position of the core promoter, the exact position of an enhancer, the exact nucleotides that control each one of these. So we do this. So we have now the luciferase gene, which is right here, the luciferase gene under control of a promoter of interest. And I don't know what the different sequences represent. Now I want to say, here we have a repressor region, here we have an activating region, and here we have the core promoter sequence. Now I don't know what the positions of these sequences are, and that's what I want to study. So I take the DNA piece that contains what I think is the promoter region containing all of these regulatory sequences. So I take it, I clone it into a plasmid. I clone it into the plasmid that contains the luciferase gene. I create a recombinant DNA here. And I take this recombinant DNA, this plasmid that contains the luciferase gene that is under control of the promoter that I want to study. Okay? And I insert it or transfect it into certain cells, nerve cells, skin cells, stomach cells, whatever. And I study how the luciferase gene is controlled by these sequences. And what I do is the following. So I take the luciferase gene, the recombinant DNA that contains the luciferase gene, and I place the promoter that I want to study. Okay, the complete promoter. Okay, this whole thing that contains the core promoter, the activating region, the repressing region or repressor region. And I start creating different fragments of this right here. So I start chopping up, I start deleting different regions. So for example, I remove the part of this region, I remove this region, I remove this whole thing, I remove this thing right here, I remove this core promoter complex, and so on. So I have, again, the luciferase gene under control of the whole promoter region, the whole thing right here. And then I start chopping up this piece of DNA. So I remove this part right here. Then I remove this part, creating this. I remove this part right here. So I have now the luciferase gene under control of the core promoter and the activating region. And I remove this part, I remove that part, I remove this whole thing, and so on. Now here I have a piece of DNA that contains no promoter whatsoever, just that luciferase gene. Now the luciferase gene is right here. So this piece of DNA right here is represented by the black arrow. Okay, now, so let's look at the results. Now I transfect, so I have different plasmids, different promoter, pieces of promoter, the same luciferase gene, nothing is changed with the luciferase gene, and I transfect these different plasmids into cells. So each group of cells would have a recombinant plasmid DNA containing the luciferase gene and different pieces of the promoter. Okay, and I measure the amount of light that is produced by by converting the luciferin uh, substrate into the light product. Okay, great. Now, I have to have controls. I have to have things that either work or do not work, so that I can compare my experimental DNA with the controls to make sure that the conditions of my experiment are fine. So I create negative controls and I create positive controls. That is, something negative control is something that I know does not work. A positive control is something I know works for good. Now, if the negative control works and it produces luciferase, it means that there's something wrong. If the positive control that should work, if it doesn't work, it means that again there is something wrong with the DNA that I've prepared. Okay, so here we have the luciferase gene with no promoter whatsoever. I know that the luciferase gene does not have a promoter. That's a negative control, something that I know does not work. And here we have the luciferase gene that is under control of a good promoter, a different promoter, a promoter that I know works great. So that's a positive control, just to make sure that the conditions of my experiment are fine. So, and that's the amount of light right here. Okay, so here we have the luciferase gene under control of no promoter. There is very little signal. So someone would say, whoa, wait a second. There is no promoter. Why is there a signal? Well, remember when I talked about leakage of promoter regions? Genes can be expressed even though they have no promoter, even though there are no activators, and so on. Well, there's little signal. So that's a negative control. That's what no expression means. Well, there is some expression, but that's what no expression means, because now the luciferase gene is not under control of any promoter. Okay, here we have luciferase gene under a good promoter, different promoter. Again, that's a positive control. There is excellent signal right here. Again, there's a lot of expression, 100%. So that's my 100%. Okay, now Here we have the luciferase gene under control of the promoter that I'm testing under certain conditions, the whole promoter. Okay. Here we have a signal. That's what an expressed luciferase indicates when under control of this promoter under study. Okay, so we have this signal right here. Okay, then what I start to do is I start removing pieces of DNA, pieces of the promoter region. So I start removing the repressor region. So what do you think would happen? If I remove the repressor region, I should see an increase in expression because now there are no repressors that can bind to the promoter and can inhibit expression of the luciferase gene. Okay, so by removing the repressor region, I should get increase in expression, and that's exactly what happens. So when under control of the whole promoter, including the repressor region, I get this amount of expression. If I remove the repressor region piece by piece, little by little, I get more expression. Wonderful. So I know that this region contains a repressing region. Okay, now let's say that I start removing, now I remove this whole thing, right? This is what I get. Now there's basically, by the way, no difference between this and that. Okay, so this is just experimental conditions. Now I start removing the activating region. What do you think would happen if I remove the activating region? I should see a reduction in expression, and that's exactly what I see. If I remove the activating region, there is reduction in expression of luciferase again. Okay, there is reduction in the amount of light that is produced. Okay, so and that's exactly what we see. There is complete reduction in expression of the luciferase gene by removing the activating region. So I know that this region contains an activating region because when we remove it, there is no expression of the luciferase gene. What would happen if I remove the core promoter? Even more reduction of expression of luciferase, less production of light, and that's exactly what I see. This here is similar to this right here, right? Because that's what no production of luciferase means. Very little production of light. Same thing here. Okay, so by removing the core promoter, there is no expression whatsoever. So this is called the luciferase reporter gene assay. The luciferase reporter gene assay. Assay means experiment that measures something. Reporter gene is a gene that gives us an idea. It reports something. And this gene is the luciferase gene, the fruit fly gene, that converts luciferin into a product that produces light. The more luciferase we have, the more light we have. So here we have an experiment where we want to identify the function of different promoter regions, different pieces of a whole promoter, whether this sequence represses, inhibits protein expression, or activates protein expression, or if it is essential for gene expression, that is the core promoter. Or we can place it inside cells, we can transfect cells, and we can put these cells under different conditions again, in the presence of hormone or no, stress or no, nutrition or no, you name it. Okay, and we can measure how a promoter a specific promoter functions, how it behaves under different conditions by measuring the amount of luciferase that is produced, which is represented by the amount of light that is produced as well. الله معكم.
خلينا نشوف example شوي more complex. تكلمنا احنا على هذا البروموتر region as an example of a gene that can be regulated by different regulatory factors, transcriptional regulatory factors. مثلا في عندنا serum response element، هذا اللي بتأثر بوجود النوترينتس. في عندنا estrogen response element، هذا بيرتبط مع الاستروجين ريسبتور في وجود الاستروجين. فالاستروجين بيرتبط مع الريسبتور، بدخلوا على النوكليوس، بيرتبط مع الاستروجين ريسبونس element، فبصير في stimulation للجين اكسبرشن whatever that gene is. الجين نفسه برضه بيكون under the regulation of the progesterone عن طريق ارتباطه بالبروجسترون ريسبتور، بدخل على النوكليوس، بصير في ارتباط مع البروجسترون ريسبونس element، فبصير في stimulation للجين اكسبرشن. طب احنا عندنا هذا البروموتر ريجن، بدنا نشوف وين هي الريسبونس elements اللي بتستجيب للاستروجين، بروجسترون، النوترينتس الى اخره. يعني هذا هو البيربس تبعنا. كثير كويس. فاللي عملنا انه اخذنا هذا البروموتر ريجن، حطيناه يعمل كنترول للجين تبع اللوسيفيريز، فبدل الجين هذا الاوريجينالي تبعه، لا احنا حطيناه اللوسيفيريز جين يكون under control of the promoter region of this promoter region. طيب فاللي عملناه هون انه بنحطهم بالبلازميد، وبنحط البلازميد جوا السيلز، بنعمل ترانسفكشن، يعني بندخل البلازميد جوا السيل. فالجين هلا under control of the different conditions بوجود الاستروجين، بروجسترون او النوترينتس. طيب فعندنا conditions، different conditions. اول شيء حطينا البلازميد again جوا السيلز، وما حطينا شيء مع السيلز، اوكي، بس يعني امور شيء عادي جدا يعني whatever that is يعني. وطلعنا على الاكتيفيتي تبعت اللوسيفيريز، فطلع لنا مثلا هي 100% لوسيفيريز اكتيفيتي. هذا اللي بنسميه basal expression. basal expression يعني the minimal expression بعدم وجود اي inhibitors او activators. اذا حطينا السيلز in the presence of estrogen، شو حيصير؟ حيصير في stimulation للجين اكسبرشن. ليه؟ لأن الاستروجين حيدخل جوا السيلز، حيرتبط مع الاستروجين ريسبتور، حيرتبط الاستروجين ريسبتور مع الاستروجين ريسبونس element اللي موجوده في البروموتر ريجن تبعت اللوسيفيريز هلا، فراح يصير في stimulation للترانسكريبشن، وبالفعل صار في عندنا زياده بالاكتيفيتي اوف ذا لوسيفيريز انزيم. طب لو حطينا بروجسترون، اوكي، حطينا هون بروجسترون مع السيلز، نفس الشيء، بروجسترون حيدخل جوا السيلز، حيرتبط مع الريسبتور، حيدخل جوا النوكليوس، حيرتبط الكومبلكس مع البروجسترون ريسبونس element، حيصير في stimulation للجين اكسبرشن، وبالفعل صار عندنا زياده بالاكتيفيتي اوف ذا لوسيفيريز جين. طب لو حطينا السيلز بكونديشن فيها نوترينتس، حيصير في استجابه انه if you remember MAP kinase signaling pathway، حيصير في activation لل serum response factors، هدول حيرتبطوا مع serum response element، وراح يصير في stimulation للexpression of the luciferase، فبالتالي راح يصير في زيادة بالluciferase activity. ممتاز. إذا هذا هو الكنترول تبعنا، هذا هو اللي المسطرة اللي احنا راح نقيس عليها أي شيء راح يصير بالسيلز. هلا اللي احنا بدنا إياه we want to confirm that this is the element that is responsible for binding to the progesterone receptor. بدنا نتأكد، نعمل confirmation إنه هذه الelement هي اللي تستجيب لوجود الاستروجين. وبدنا نتأكد إنه هاي الإليمنت هذا الريجن of the DNA اللي تعمل كنترول للإكسبريشن of the gene لما يكون في نوترينتس أو ما في نوترينتس. ماشي؟ طيب فإيش عملنا؟ أخذنا البروموتر ريجن وشلنا الاستروجين ريسبونس إليمنت، هذه شلناها. أوكي؟ جينيتيك انجينيرينج. حطيناها جوا السيلز، البلازميد جوا السيلز، فطلعنا على اللوسيفريز اكتيفيتي، فهذا البيزل اكسبريشن تبع اللوسيفريز. ماشي؟ حطينا الاستروجين على السيلز. لاحظوا انه هون لما يكون الاليمنت موجوده قد ايه الاكتيفيتي اوف ذا جين حتكون عاليه. لكن لما شلنا الاستروجين ريسبونس اليمنت يعني زيادة طفيفة جدا، يعني it's nothing. معناته هذا confirmation انه هاي الاليمنت هي اللي بتستجيب لوجود الاستروجين. لانه لما نحط احنا بروجسترون مع السيلز اللي فيها البلازميد اللي فيها البروموتر اللي ما فيهوش الاستروجين ريسبونس اليمنت، صار في زيادة. معناته انه البروجسترون ما بيشتغل عن طريق هاي الاليمنت. طيب ولو حطينا السيلز بنوترينتس برضه في ستيميوليشن للجين اكسبرشن. هلا ما يغركم النقصان اللي احنا شايفينه هون او الزياده اللي احنا شايفينه هون، لانه هذه يعني experimental error. لكن لما نشوف انه كان تقريبا 180% نزل لتقريبا 110%، this is huge difference. طب شلنا البروجسترون ريسبونس element. بهاي الحالة نتوقع انه البروموتر ريجن ما راح تستجيب والجين اكسبريشن ما راح يتغير بوجود البروجسترون، لانه الاليمنت اللي بيشتغل عن طريقها راحت. لكن الاستروجين حيشتغل والنوترينز وجودها راح ياثر على الجين اكسبريشن. وبالفعل هذه السيلز بدون اي شيء، هذه السيلز اللي فيها البروموتر ريجن اللي ما فيهوش بروجسترون ريسبونس اليمنت. طيب فالبيزل اكسبريشن زيه زي هذا وزيه زي هذا. ممتاز. لما حطينا الاستروجين لاحظوا الزياده اللي صارت بالاكسبريشن تبع اللوسيفريز. ليه؟ لانه هذه الاليمنت موجوده وبالتالي الاستروجين اشتغل. لما شلنا هاي الاليمنت وضفنا بروجسترون ايش راح يسوي؟ ولا شيء، ما راح يشتغل، لانه الاليمنت اللي هو بيشتغل من خلالها راحت. فاحنا عملنا هيك كونفirmation انه هاي الاليمنت هي الاليمنت اللي بيشتغل فيها الاستروجين البروجسترون. ولو حطينا نوترينز برضه في زياده، راح يصير في افكت على الجين اكسبريشن تبع اللوسيفريز. طيب شلنا السيرم ريسبونس اليمنت. again هذا البيزل اكسبريشن بدون وجود اي شيء، زيه زي بقيه الكونديشنز اللي موجوده عندنا هون. حطينا استروجين صار في زياده، لانه الاستروجين حيشتغل عن طريق الاليمنت تبعته. بروجسترون برضه راح يصير في زياده بالاكسبريشن، لانه الاليمنت اللي بيشتغل من خلالها موجوده. حطينا النوترينز ما صار في اي تشينج بالاكسبريشن تبع اللوسيفريز، لانه هاي الاليمنت مش موجوده، وبالتالي النوترينز ما راح تاثر على الاكسبريشن اوف ذا luciferase جين. فاحنا ايش عملنا هون؟ عملنا confirmation، اكدنا انه هاي الاليمنت هي اللي بتشتغل عن طريقها النوترينز، هاي الاليمنت هي اللي بيشتغل عن طريقها الاستروجين، هاي الاليمنت هي اللي بيشتغل عن طريقها البروجسترون هرمون. فهيك احنا again عملنا identification ل the different transcriptional regulatory binding sites, protein binding sites. والله معكم.